So let's start with the meiosis. I don't know where are the rest of the people. So do you have any doubt in this one? Mitosis? Tell me, Martin.
No, sir.
Everything is clear to you?
Well, I mean, yeah. So
That's great.
you need to
So
explain that big paragraph.
yeah, if there is any doubt, tell me. I can explain for that. Any doubt?
No, sir. No. The meiosis part, yeah, please.
Okay, fine. So let's start with the meiosis. First, I will let you understand because the understanding is more important than writing. Because whatever we understand, that not that we have to explain it, right? So meiosis and mitosis, these both are the basically the processes where you can understand each and everything without writing even a single letter. But yes, we have to write it in our school examination. So I'll dictate you as well. So first, just put the heading meiosis. So, meiosis is a big process. It's a lengthier process than mitosis. Fine. I'll explain one by one. So look, the second phase of we divide meiosis into two parts, right? Meiosis first and meiosis second. Second part of meiosis is exactly same as we have studied in mitosis. But the first part of meiosis, it is a lengthier, means it take much time, and it is a bit complex as well, right? Look, so this is interphase nucleus. In interphase nucleus means now the nucleus will start or enter into, or rather than nucleus, now the cell will enter into division process, means M phase. Out of this M phase, this time it will go for meiosis rather than mitosis. So mitosis is a general cell division which takes place in every part of our body. For sake of example, if my skin get damaged, then the cells that will replace the damaged cell or dead cell, the new cell will divide and they will fill that gap. The process which is going to take place, that will be the mitosis. Second, if the new cell have to be formed, then the process will be mitosis. But where the gamete formation take place in the body of any plant or animal, gamete formation means sperm and egg, right? So basically the gametes are haploid. So wherever the gamete formation takes place, we call that process gametogenesis. So wherever the gametogenesis will take place, the process will be meiosis. So basically in the body of every plant or animal, the process which take place, that is meiosis, where this process takes place only at the gamete formation or wherever the gamete formation is taking place. For sake of example, if we talk about the human, the meiosis takes place in gonads. Gonads means where the sperm or egg is being formed, right? Rest of the body generate new cells, or rest of the body, wherever the cell division takes place, that is a mitosis. So meiosis takes place during gamete formation. Now, this is interphase cell or interphase nucleus. Here, the number of chromosomes is 46, means we are diploid. Now, in meiosis first, these are the centrosome, and this is the nucleus and nuclear envelope. What will happen in prophase first? So in prophase first, this nuclear envelope start degenerating or disintegrating. The same way, the centrosome will divide and they will start moving towards the opposite pole. Now, what will happen at this point of time in prophase? The two chromosome comes together, right? Remember, in mitosis, what happened? There is a single chromosome, so we call that monovalent. Here, two chromosome come together in pair. We call this structure bivalent. So, in prophase of meiosis, bivalent formation takes place, while in prophase of mitosis, only monovalent formation takes place. That is the first difference between the meiosis and mitosis. Now, what will happen in meiosis? These the spindle fiber formation will take place in prophase first step. Spindle fiber formation start, and these structure are called ester rays. Ester rays formation takes place. Again, in metaphase, what happened? The chromosome or bivalent, they arrange themselves at the center or the equator. We call this structure metaphasic plate, right? Spindle fiber which are arising from the centrosome, they will attach with the kinetochore of the or centromere of the chromosome, right? Now in anaphase, what will happen? They will be pulled apart, means they will be pulled apart. They will move toward the opposite pole. Before moving ahead, I want to tell one thing. You can see here, there are one type of the chromosome, they are red in color, and another have been shown in the blue in color, right? Means they are not sister chromatid. They are non-sister chromatid. Means both are the different chromosome. Here you can see in metaphase, some part of the red and blue, they have exchanged from each other. Can you see, Martin? Is it visible to you?
Yes, sir.
They are mix
Yeah.
of blue and blood. That is called crossing over. They have exchanged some fragment of each other. They have exchanged the fragment. And exchanging the fragment is called crossing over. So in meiosis first, or in a meiosis, crossing over takes place, while in mitosis, crossing over do not take place. Second difference. Is that clear, Martin? Clear?
Yes, sir.
Okay. Now what will happen? Anaphase first. After anaphase, there is a telophase. Sometime telophase takes place, sometime telophase do not takes place. Here, two cells get separated, but before they getting separated, they have exchanged their part. Now you know, here both of the cells, they are haploid, they are not diploid. So from one cell, two haploid daughter cell have been formed, right? Cytokinesis may takes place, sometimes cytokinesis do not takes place. Now from here onward, right, the meiosis second start, right? Meiosis first is called reductional division. Martin, I want your 100% focus over here. This is very important point. Meiosis first is called reductional division. Why? Because from a diploid daughter
Divided.
cell, a diploid mother cell, formation of diploid daughter cell takes place. Clear? Right? But here now these two cell will become mother cell, and they will form the four cell. All cell will have this mother here. Both of the mother cell is haploid, and four daughter cell is haploid. So meiosis second is called equational division, where mother cell and daughter cell have equal number of chromosomes. So here mother cell is haploid. Now what will happen? This stage is exactly similar to the mitosis. In prophase second, again the nuclear envelope get dissolved or disintegrate. Both of the centrosome will start moving toward the opposite pole. In metaphase second, both of the cell will have or their chromosome at equator. We call it equatorial plate. Again, in anaphase second, they will be pulled apart, means they will break down from the centromere at the mid. And in the telophase, formation of four daughter haploid cell takes place, right? And then the cytokinesis takes place. So finally, in the process of meiosis, from single diploid mother cell, formation of four haploid daughter cell takes place. This complete process is called meiosis. Tell me, Martin, if you have doubt at any point.
The can I draw like the two and like
Uh,
division?
sorry, I didn't got it.
Can you draw it? Like, can you like like show like the reduction and equational division? Like, can you,
Achha, okay, okay, right, right. Mhm. Look, what happened? So meiosis, right? So in meiosis, there is a this is a cell, and this is a 2n or diploid mother cell, right? Now what happened? So in diploid, why I am saying, so there are diploid means 23 plus 20. Clear? These 23, so try to understand. Their number of the chromosome is 23, but they have, every chromosome have the DNA double DNA. So number of the chromosome is equal, but their DNA content is double. Did you got it or not? There are 23 such kind of the chromosomes. Got it or not? Tell me, Martin.
Yes, sir.
Okay. So this kind of 23. Now, what will happen from a diploid means given from a diploid mother cell? So what is the number of chromosome over here, Martin? Number of chromosome is 23 plus 20, right? So total 46 chromosome in mother cell.
Yeah.
Now the formation of two daughter cell have taken place. Now each daughter have 23 chromosomes. So this is the
So 46 divided into 23.
right daughter cell. This is the second daughter cell. Clear, Martin? So look,
Yes, sir.
this is the meiosis first. So number of chromosome have reduced to half. So can I say it reductional division or not?
Yeah, yeah.
So that's why meiosis first is called reduction. Clear? Now look, these daughter cell which have, they have 23 chromosome, but the chromosome content is equal to 46. Their chromosome content is 46. Got it? Now what will happen? These daughter cell will divide. Now these daughter cell will become mother cell. Got it? Now they are mother for the next one. Yes, Martin, clear or not?
Yes, sir.
Now what will happen? The chromosome will break from here, right? Now it will form two daughter cell having the 23 chromosome. Clear? Second daughter cell, it having the 23 chromosome, right? The same way will happen over here. Now what is? Oh, 23 chromosome. This one. 23 chromosome, right? So look here. Now again for the meiosis second. For meiosis second, this is the next mother cell. Yes, Martin. Now this daughter cell have become mother cell for the meiosis second. So mother cell have 23 chromosome. Now daughter cell have equal chromosome to mother. Clear? So can I say meiosis second is equational division? If you want, you can draw it. Is that clear? Everything clear to you? Pardon? Now?
So yes, sir, like which one is haploid and which one is diploid?
This one is haploid. This is the haploid because they have the n number of chromosomes. N 23 only 28. Here they are the 2n, so diploid. So this is haploid. This is also n number of chromosome. Haploid, 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 haploid. Got it or not? Tell me.
Okay, so like which one is diploid?
This one haploid. This is haploid. They all n are haploid. Only diploid. This is the only diploid. Rest of all are haploid. So draw it, Martin, right now. If you understood everything. And even if you have any doubt, ask. Feel free to ask.
Yeah, done, sir.
Done. So
Wait,
note it down.
I forgot to write.
Run it. Draw this diagram, this one, and then leave the space for the diagram which is visible to this one. Right. You have to draw this diagram in the after the class, because when in the exam, if you get the question, you have to draw this diagram. Even you can draw this one as well.
Okay, now
Done. Okay. Now, before going into deep, one more important thing. Here, this crossing over process, which takes place in prophase first, this is not that much simple. Further, meiosis prophase one have been divided into these stages. They have been described in your this one NCERT as well, right? So now try to understand part. Further, meiosis first have been divided into these stages: leptotene, zygotene, pachytene, diplotene, and dikinase. You can see leptotene, zygotene, pachytene, diplotene, and dikinase. They have been mentioned in your NCERT book, page number 167, 168. Look, in leptotene, what happened? Nuclear membrane, try to understand. In leptotene, what happened? Nuclear membrane dissolving degradation starts. Now the chromosome condense and they start making bivalent, right? Now next stage, so here the bivalent formation I have not taken, but yet the in leptotene bivalent formation I have not. Nuclear membrane is there, right? And these chromosomes start condensing, means they start becoming thicker. In zygotene, what happened? Bivalent formation takes place. What is bivalent formation? Two chromosomes, they come together to each other, right? And some part of chromosome get attached with the sister chromatid of another chromosome. This complex where the attachment takes place, this complex is called synaptonymal complex formation. So synapses, and this process is called synapses. Try to understand. This structure is called synaptonymal complex, and this process called synapses. So synapses takes place in zygotine stage. What is synapses? In synapses, two arm of non-sister chromatid, they attach with each other and form a complex. This is called synaptonymal complex form, right? Clear, Martin? Synapses and synaptonymal complex, clear?
can you say again, sir?
Yeah, sure. Look, when two non-sister, they are non-sister chromatids. Look, try to understand. Here I have shown it. You can see this chromosome is black color and green color, two different chromosomes. This chromosome, the arms are these, both arms are black, so they both are sister chromosomes. Clear? Look, here both arms are different. These green are sister chromatids, right? But black one and green one, they are called non-sister chromatids. Clear?
Yes, sir.
Okay. Again, I have shown here. 1, 2, 3 genes are arranged in the same way. Both exchange each other. So they are called non-homologous chromosome, right? Non-homologous chromosome, they exchange parts of each other. This exchanging part process is called crossing, right? You can see in pachytene chromosomes, these are the chromosomes. These chromosomes come closer to each other, right? Bivalent formation have taken place. Now, where the synaptonymal complex have taken place, they have exchanged the part of each other, or they have rather part, they have exchanged genes with each other. Bivalent form and crossing over take place. This exchanging the part is called crossing over. Clear? The process of exchanging part of each other is called crossing over. Clear?
Yes, sir.
Now come to diplotin. In diplotin stage, what happened? Chiasmata formation takes place, right? Synaptonymal complex, they dissociate. What is the synaptonymal complex? Here we have seen the synaptonymal complex. They try to separate from each other after exchanging the part. Then they form the scissor-like structure, this crisscross structure, right? This crisscross structure is called chiasmata, right? Chiasma or chiasmata. So synaptonymal complex dissociate, and in diplotin, chiasmata formation takes place. Clear?
Yes, sir.
So bivalent clear, synaptonymal complex clear, crossing over tier, and chiasmata. And finally, in dikinesis, the nuclear membrane fragment they dissociate, right? And here the chromosome which have exchanged their part get separate, and this is called end of the prophase. So these are the stages of prophase: leptotin, zygotin, pachytin, diplotin, and dikin. Go through with it and tell me. That's why I said that my meiosis is lengthier process, and this is the complex in comparison to mitosis.
Yeah, okay. Understood.
Understood?
So like the end of the chromosome
This,
they like combine.
this is the end of prophase. This is the end of prophase actually, right? Not end of meiosis. So look again. I will go back. Look here. This prophase. This process have been divided into four parts: diplotin, zygotin, diplotin, dikinesis. Got it, Martin? Clear or not?
Yes, sir.
Okay. So fine. Start writing down the meiosis. Put the heading meiosis. Start writing down first. What is meiosis? Meiosis is a double division. Meiosis is a double division which occur in a diploid cell, which occur in a diploid cell or nucleus, and gives rise to four haploid cells where each cell where each cell having half the number of chromosome, half the number of chromosome as compared to parent cell, as compared to parent cell. We have started actually class at 6:30, right? You people are too late. If I will repeat it, not 6:30, yeah, so almost you are 36 minutes late. So if I will repeat it again, then it will again take too much time, right? So you can do one thing. You can take the link of video and go through with it after the class. try to understand. And in case if you are facing problem, in next class you can ask them, right?
Okay, sir. Sir, I was having exam. Because of that, I couldn't join, sir. Sorry.
Okay, yeah, I have seen you after long time.
Yes, sir. I was having exam. So sorry. Sir,
No issue.
only this picture is enough, right?
No, this picture is not enough. Basically, this is the meiosis. So, meiosis takes place in two parts. This upper part is meiosis first, and lower is meiosis second. Then further, this prophase one is the longest phase. This prophase one have been further divided into five different stages. So, this meiosis prophase first, I have written over here. Meiosis prophase first have been divided into five stages: leptotin, zygotin, pachytin, diplotin, and dikinase. Right.
Sir, the
So,
link of this I don't have, sir. Can you just share it, sir?
no,
Because
actually,
last time the management shared, but that wasn't the updated one. That was
okay,
so different from what you're teaching.
you can do one thing. You can message them. Please provide me the link for the today's class. Drop the date of today, right? Then they will come to know. Fine, because the management team, the team which is operation team. So they are the people who can share who have access to sharing the link, right? I don't have access to
Yes.
share the link, right? So
Okay, sir.
they have your number, they have your group, right? So they can share you. Fine. So if you'll drop the message to the person who's in your contact from our team, they will give you. Yeah. Martin, are you saying something? Yes, Martin.
I didn't say like, sir. I didn't say.
Okay, fine. So, uh, okay. So, start writing down. And that was the definition of meiosis. Fine. And this is very, very important. If in any exam they ask you the question, full mark, 5 marker question, definitely it will be from meiosis. And even in medical exam, the question which comes from this chapter, they are mostly from the meiosis. Right. 99% they are from meiosis. 95% they are from 5% they may be from cell cycle or they may be from meiosis. Fine. So this was the definition of meiosis. Now next thing, put the heading meiosis first, right? Meiosis first subheading. So main heading was meiosis. Second subheading is meiosis first. Start writing now. Meiosis first is called reductional division. We have discussed in detail, Martin. Meiosis first is called reductional division. Here I have discussed already. Meiosis first is reductional division. Full stop. It can be studied under 4 stages. Prophase first. It can be studied under 4 stages. That is prophase first, comma, metaphase first, comma, anaphase first, and telophase first. Now, under this subheading meiosis first, there is again subheading A. That is prophase first. Prophase first. It is a complicated prophase first. It is a complicated and prolonged phase compared to similar stage of mitosis. Full stop. Prophase first is divided into prophase first is divided into 5 subphases. 5 subphases. Leptotin, comma, zygote, comma, pachytene, comma, diplotin, or dikinesis, or dikinesis. So meiosis first, this prophase first have been divided into further these substages. Now start writing down. First, leptotin. In leptotin, nucleus enlarges, full stop. The chromatin fiber becomes short and thick. Chromatin fiber becomes short and thick to take form of chromosome. Full stop. They are called homologous chromosome. And next substage, zygote. Next substage is zygote. In zygote, the two chromatids which belong to non-sister chromosome they come closer to each other and form synaptonymal complex. Give me 1 second, guys. Just one. Yes. So where I was, yes, synaptonymal complex, and the process is called synapsis. Next point, pachytene. In pachytene, the two chromatids belonging to same chromosome called sister chromatid. Full stop. The chromatid belonging to different chromosome are termed non-sister chromatid. Full stop. The non-sister chromatid exchange the part or genes. Non-sister chromatid exchange part or genetic material The process is called crossing over. The process is called crossing over. Full stop. Next, diplotin. In diplotin, the synaptonymal complex in diplotin, the synaptonymal complex, the complex which formed over here, this synaptonymal. In diplotin, the synaptonymal complex start dissociating and non-sister chromatid and non-sister chromatid separate from each other and non-sister chromatid separate from each other. Full stop. The crisscross structure formed during this stage is called synaptonymal complex. Sorry, so it's called chiasmata. The spelling is written over here. Chiasmata. Chiasmata. Next is dikinesis. In dikinesis, nuclear envelope start dissociating. In dikinesis, nuclear envelope start dissociating and fragmentation takes place. This process is called This process is end of prophase. So this was the meiosis prophase 1. Is that clear? Martin and Shadika, clear? Note it down.
Sir, can you explain the process?
Which one?
No, like how it's transforming. Can you just say it like in
Yeah.
can you just explain, sir, without
Yeah.
like
In liptotin, you can see this is the chromosome. So chromosome condense, they become thick. So you can see this blue have 2 part and red have 2 part. these I have explained over here. These both part, like in the same way, this black have two part and green have two part. These both have the arms. So this arm and this arm. These both arms are called sister chromatid, right? Again, for the green, these both arms are sister chromatid, but one arm of black and one arm of, so you can say one chromatid of black one and one chromatid of green one. They are non-sister chromatid for each other. Got it? So here you can
Okay,
see
sir. For each other, they are non-sister chromatids.
exactly for each other they are not sister chromatid, non-sister chromatid, right? So as
Yes, sir.
here, like this 1 and 2, they are sister chromatid for each other. This is 1 and 2. So 1 and 2 are sister chromatid for each other, right? 3 and 4, they are sister chromatid for each other. 3 and 4, they are sister chromatid for. But 2 and 3 are non-sister chromatid for each other. Got it? 4 and 1 are non-sister chromatid for each other. Clear? Because they are part of different chromosomes. Fine?
Yes, sir. Yes, sir.
So here you can see in zygotine, they come closer to each other, and some part of non-sister chromatid they attach with each other. Here they have attached with each. This structure is called synaptonymal complex. Now here you can see in packetine, the synaptonymal complex, they have exchanged part of each other. So blue one have taken the part of red one, and in exchange it have given the blue part to the red one. So they have exchanged part, or they have exchanged genes from each other. This process is called crossing over, right? Exchanging of part. After that, in diplotin, they start separating from each other. When they start separating from each other, they make Caesar-like structure. This Caesar-like structure is called chiasmata. And in dikinesis, both of the non-sister chromosome or non-sister chromatid, they separate from each other and nuclear envelope dissolve. So this process is called dikinesis. Clear?
Yes, sir.
So this
Clear, sir.
complete process, prophase 1, meiosis prophase 1. This is the longest phase.
Yes, sir.
Now come to the, again, metaphase 1. So this is simple stage. So put the heading, again, the second subheading, metaphase 1. In metaphase 1, the bivalent chromosome, the bivalent chromosome arrange themselves on equator of the bivalent chromosome arrange themselves on equator of bipolar spindle, on equator bivalent bipolar spindle. Next point, each chromosome get attached, each chromosome get attached to a spindle fiber at centromere. Next, anaphase, next subheading, anaphase first. In anaphase first, this is phase. The homologous chromosome, break their connection. The homologous chromosome break their connection and separate out. Full stop. The process of separation, is named as disjunction. The process of separation is known as disjunction. Next point. The chromosome moves towards the pole with the help of the chromosome move towards the pole with the help of tactile spindle fiber. Tactile means they are means contracting, right? That's why they are tactile fiber. Next, telophase first. This one, telophase first. A nucleus is formed again as nuclear envelope reappear. Nucleus is formed again as nuclear envelope reappeared. Right? And further, cytokinesis takes place. Is that clear?
Sir, can you explain this also?
This one here, look, it's very simple. It's like mitosis. There are centromere, there are centrosomes. Centrosome gets separated, they make fiber-like structure. They are called spindle fiber. They are the diode formation takes place, prophores. Then what happens? These chromosomes get arranged themselves at the equator. We call it equatorial plate. These are the centrosome, which form fiber-like structure. They are called spindle fiber. Spindle fiber get attached with the chromosome, and this is called metaphasic plate when they arrange at the equator. And in anaphase, what happened? This spindle fiber start pulling these chromosome apart at the opposite pole. We call it anaphase. And finally, here in telophase, both of the spindle fiber reaches at its pole, and around them, the formation of nuclear envelope takes place. So this is meiosis. Put a note. Note. This is for all of you. This note will be a question. Meiosis first is called, I have explained the same. Meiosis first is called reductional division. Meiosis first is called reductional division because meiosis first is called reductional division because the number of chromosomes get half. The number of chromosomes get half as the mother cell is diploid, but as the mother cell is diploid, but both of the daughter cells are haploid. Tell me, all of you, is that clear everything? Till meiosis first, then meiosis second
Yes,
is
sir.
easy. Okay, fine. So meiosis second is easy. It's like mitosis only, right? So next put the subheading meiosis second. Meiosis second. Meiosis second is shorter than a typical mitosis. Full stop. The division maintain the number of chromosomes. That's why it is called equational division. Look here, after the meiosis, first both are the haploid set, right? Haploid daughter cell, 23, 23. Now these daughter cell will become mother cell for next division, right? Now you can see here both of the cells have 23 chromosome and their daughter also have the 23 chromosome. So number of the chromosome is equal in meiosis second in mother cell and daughter cell. That's why we call it equational division. But here in meiosis first you can see mother cell have diploid but daughter cell are haploid. So here the number of chromosome have reduced. That's why it is called reductional division, right? So meiosis second is called equational division and meiosis first is called reductional division. This may be the question. Normally what they will ask you in the question, justify that meiosis first is a reductional division but meiosis second is equational. Then you have to make this diagram simply and you have to write it down the complete process, right? So meiosis second can be further meiosis second can be further divided into prophase second, comma, metaphase second, comma, anaphase second, and telophase second. So first head subheading is prophase second. In this stage of nuclear division, in this
Which
stage
phase
of
is this? Which phase?
prophase second, subheading is prophase second, right? This stage of nuclear division is very brief. It takes place
It is
simultaneously.
very what? It is very
Brief, very small. Brief is very brief. Full stop. It takes place
Sir, can you repeat? It is very
brief. BRIEF. Brief is very small.
brief means small.
Very small, very small. Very brief means very
Okay,
small.
sir.
Right. It takes very short time. It takes place simultaneously. So prophase second will take place in both of the cells. It takes place simultaneously in two cells. Full stop. Nucleus and nuclear envelope. Nucleus and nuclear envelope degenerate. Full stop. Right. Then metaphase second. Metaphase second. In metaphase second, the chromosome arrange themselves. The chromosome arrange themselves at equatorial plate. Equatorial plate. Full stop. each chromosome get connected with spindle fiber, full stop. Then anaphase second. Centromere of each chromosome, centromere of each chromosome divide into two, so that there is one centromere for each chromatid. One centromere for each chromatid. Full stop. Two chromatid. Now sister chromatid are getting separated, right? Two chromatids of a chromosome separate completely. And are called daughter chromosome. Full stop. Daughter chromosome reaches at opposite pole. Full stop. Each
What can you
group,
repeat?
yeah. What else? What was the last sentence? Tell me, Martin, what you read in the last, what is the last sentence? Actually, I am just framing the things. Tell me, Martin, what was the last sentence?
They go to the top spin. They go to the from there.
They go to opposite pole. They go to opposite pole, right? Full stop. Each group have haploid number of chromosomes, right? Next is telophase second. The four group of chromosome, here there are four groups, 1, 2, 3, 4, right? So four group of chromosome arrange themselves into haploid nuclei. Full stop. Each cell form a nucleus full stop. Spindle fiber usually degenerate and further protoplasm divide. Full stop. So this was all about the meiosis. Still, if you have any doubt in meiosis, you can ask me. Then I will dictate you the significance of meiosis, because significance of meiosis is also very important. Why the meiosis takes place? Look, what is the purpose behind meiosis? During gamete formation, meiosis takes place. During sperm formation or egg formation, meiosis takes place. So every cell of our body have 46 chromosomes, but sperm have or egg have only 23 chromosomes, right? Why? Because in next generation, when sperm and egg fuses, each have 23 chromosomes and they become 46. If meiosis will not take place, then we have the 23, we have two pair of 23, right? So parents have the 23 pair, 23 pair. If they will not get separate, means meiosis will not take place in next generation, there will be 4 set of chromosomes, 4 set of 23. In next generation, there will be 8 set. In next generation, there will be 16 set. So after every generation, the sets of chromosome will increase and a human will become a giant, right? That's why meiosis is very important, so that after every generation, the number of chromosome remains same. Clear or not? Clear? Martin, Shritika,
Yes,
all
sir.
of you? So the next thing is, please write it down. Significance of meiosis. What is the significance of? The first significance, first point, write it on bullet form, because you can get this question as a 2-marker question or 3-marker question. What is the significance of meiosis? So please write down. First point. Meiosis form gamete. Meiosis form gamete, which are important for meiosis forms gamete, which are important for sexual reproduction, which are important for sexual reproduction. Next point. Meiosis maintain fixed number of chromosome. Meiosis maintain fixed number of chromosome in sexually reproducing organism. Next, crossing over make new combination. Crossing over make new combination of genes. Fine. So this was the significance of meiosis. Now I have a question. There are a parent, means a father and a mother. A very practical and very logical question. So there is a parent, means a father and a mother. They have 4 children, right? 2 boys and 2 girls, ideally. But you will see across the world, all those 4 child will have different faces, different body structure, different features. How does it possible? At least why all the kid of same parent do not resemble or matches with each other? Is it there or not? Until unless they are not twins, they do not resemble.
Variation. Because of variation.
Right. Very good, Shritika. How does that variation takes place? That's what I want to know. How does it possible? How does this variation takes place? Look, during gamete formation, crossing over takes place. During gamete formation, crossing over takes place. So every time whenever the crossing over takes place, every sperm or every ova will have different set of chromosome because of the crossing. So because of the crossing over, new combination is formed. That's why the children do not match it with their parents because of the crossing over. Got it?
Yes, sir.
So variation comes from crossing over during the gamete formation. Fine. That's why all the sisters or cousins, they do not match it. Else, other if crossing over would not be there, then the all the children of the same parent will have the same faces. And from the one gene which make the face, you can make at least 1 lakh copy after mixing them. Right. That is quite possible. Okay, so this was all about this chapter of the second chapter, that is the cell cycle and cell division. And go through with this complete chapter and ask me if you have any doubt. Then we will start with the biomod. Go through with it. Quick. Go through with it, all of you, and ask me if you have any doubt. Then we will go ahead. Yes, Indika Martin. Are you going through with it? Indika Martin and
Yes,
Priya.
sir. 1 second.
Yeah, sure. Go through with it. And even after the class, go through with it rigorously because again and again because this is a bit complex. Like mitosis is okay, but meiosis is a bit complex process.
It's clear, sir.
Okay. Martin, what's for you? Yeah,
That answer.
done? Clear? Yeah, is it clear?
Yes, sir.
Yes.
Even after the
Yes,
class,
sir.
please go through with it, right? And revise it in I think yeah, in next class, we'll start with the biomolecule part, right?
Yes, sir.
Biomolecule is yes, it is important for your school examination. And basically, out of this biomolecule, what exactly they ask? They ask in a school examination. Normally, they ask you from the uh, this one enzyme part, right? And enzyme part that is given in the last of this one, right? Uh, Chemical reaction of enzymes, right? This one and this one, the nature of an action of enzyme, right? What is the cofactor and all that, right? This kind of the question they ask, and sometimes they do ask what are the monosaccharide, disaccharide, or polysaccharide. Give some example. That kind of question. And this part is not that much very